Hey everybody, in this video we are going to talk about genetic engineering of humans. Not that I am saying it is ethical or legal, but it is possible, and we will see how it is possible in this video. So let's start with a primary oocyte. Primary oocyte. Okay, so here's the nucleus of the primary oocyte. We're going to call that the zona pellucida. Okay, primary oocyte is going to go through meiosis 1. And that's going to give us the secondary oocyte with one polar body. I'm going to put the polar body right there. And here's the nucleus of the secondary oocyte. So this is what we covered on the first day. I guess second lecture of the course. We're not keeping track of the chromosomes here. Just know that, half the chromosomes went into the polar body and half are still here in the nucleus of the secondary oocyte. So meiosis 2 commences in the secondary oocyte, but it doesn't complete until fertilization. So the secondary oocyte is released during ovulation, and it's paused in meiosis 2. So there's the first polar body there still, and I'm going to draw the spindle here showing it's paused in meiosis 2, and it's waiting for fertilization to finish. Okay, so if fertilization doesn't occur, then I guess meiosis 2 never completes. So in this diagram, I'm going to show fertilization occurring. And because fertilization occurred, so we have the little sperm head breaking through the zona pellucida. And okay, so meiosis 2 completes. I'm calling this fertilization, this step fertilization. Meiosis 2 completes, and then we have the nucleus here. This would be the maternal nucleus. Okay, with two polar bodies, the first and second polar body there. Now the sperm here has a nucleus, and both of those nuclei will occur at the same time in the fertilized egg here. And we're going to call these, I'll call this let's say, the sperm nucleus, E for egg nucleus. These are pronuclei. So they exist as membrane-bound pronuclei, before the chromosomes from the paternal parent and the maternal parent are sort of mixed together. But eventually, these don't fuse, but the membranes break down. I can diagram that like this by showing little dashed lines for the membranes. So the nuclear membranes break down, and the chromosomes can mix, which is important because they need to align now. On the metaphase plate to go through mitosis, right? So DNA would need to be replicated, and then they align on the metaphase plate, and okay, they can go through the first meiosis. And this is going to be the zygote right here, Once those pronuclei membranes disappear, you have intermixing of the genetic material. Well, that's going to be the zygote. Now the zygote's ready to undergo mitosis. And notice how I don't show the paternal and the maternal homologous or homologs lining up side by side, right? That only occurs during meiosis. Here they're lining up one on top of the other. Okay, oh, and I have to draw that zona pellucida still. And at some point we're going to lose those polar bodies. I mean, maybe something interesting happens with those, but as far as I know, I think they just disintegrate at some point. Okay, so we have our zygote here. Now, once the zygote is formed, it undergoes a process called cleavage, and that's where you have many cell division cycles without the cells enlarging, without the cytoplasm growing. So you essentially, the volume occupied by this zygote right here doesn't change with each cleavage. So the first one would look something like this. After the first cellular cleavage, we'd get the 2-cell stage. And we usually have one cleavage event about once per day for the first couple of days. And I think that's mostly in mammals, including humans. So we have a 2-cell stage right here. And I can put a polar body right here. Again, I think there should be 2 polar bodies, right? Or do they fuse? I have no clue. I think these are gonna, okay, I'm gonna stop talking about the polar bodies because I know very little about them, but I think we're gonna lose them pretty soon after the 2-cell stage. Okay, so another cleavage event gives us the 4-cell stage. Another one will give us the 8-cell stage. Then another one will give us the 16-cell stage. Again, here's the zona pellucida still here. And do I need to diagram all 16 of these? Should all be about the same size, right? I lost track, so let's assume that's 16. And this is called the morula. This is after like 3 to 4 days right here. So the morula, I believe, is a Latin term for mulberry. I guess it kind of looks like a mulberry. After 5 days, zona hatching occurs. Essentially, the zona pellucida breaks down, and then some of these cells in here start to form a membrane. And the zona hatching is kind of misleading because it makes it seem like these are like hatching out, but the zona pellucida, it's thought it actually just breaks down. Now, we're not going to talk too much about anything after this morula stage. This is really important right here. Again, this is the 16-cell stage. And this is a point at which, one can do a lot. Well, we can get genotype information of this early stage embryo, of this individual at the morula stage. And how does that work? Well, let me see. At the morula stage, there is a technique. Of course, this is not happening inside the human body. This would have to be, let's say, something that's an embryo that results from in vitro fertilization. So this is an embryo developing in a petri dish. And it is possible, using highly sophisticated equipment, is to use 2 pipettes, 2 little pipettes used for manipulating liquids and cells, specifically suction pipettes. So with one pipette, you will apply light suction to hold the early embryo in place, the morula in place. And then I could probably make these a little bigger. And this is the 16 cell stage. And I should say that each of these is called a blastomere. So each cell that results from cleavage is called the blastomere. Now, so on one side we hold the embryo in place. I've never done this myself, but a reproductive technologist or physician would do something like this. So, and over here, there's another suction pipette. It's possible to apply enough suction to gently suck out one of these blastomeres. So you can totally remove one of these blastomeres. And you know what's in the blastomere? It's going to be a nucleus, right? What's in the nucleus? Chromosomes, DNA. And apparently this embryo would be totally fine without when it loses that blastomere. You know, it can develop fine. It's like it doesn't even notice the blastomere is missing, which is pretty neat. You can lose 1/16 of your cells and it doesn't even matter. So what can we do with this, or what can a reproductive physician do with that information? Well, we can do a lot of things. So we can determine the genotype. So what can we do with the DNA from a blastomere from the morula? Well, we can determine the genotype, and we might want to do that. Let's say if you had two parents who were carriers for a severe genetic disorder, maybe a lethal genetic disorder where an infant would die in the first month, or maybe not even survive to be born. It would be possible to genotype this embryo based on the DNA in this blastomere to see if, you know, the embryo is homozygous mutant for that genetic disorder. And if it was, well, then they wouldn't implant that embryo. They would choose a different one that was heterozygous or homozygous wild type. What else is possible? How about SNP profiling? So we had a whole lecture on SNP profiling and 23andMe and Ancestry.com and those technologies, where these technologies are still in early stages, and we can't quite predict we can't get lots of phenotype information from SNP profiles yet, but we can get some information, and as we move forward, it's going to be possible to get more and more information from our SNP profiles and have more accurate predictions of the phenotype of an individual with a specific SNP profile. We can take that a step further and do genome sequencing. So not just SNP profiling, but determining the whole sequence of the genome from this early embryo, of this individual here at this very early stage. And as our ability improves to predict phenotype from SNP profiles and genome sequencing, not just, phenotype for all human traits that are determined by genotype. As our predictions become more accurate, it might be possible to, predict what this human will look like 20, 30, 40 years, what they'll be like, what their personality will be like. All of these things might be possible. Again, these are very early technologies, but somewhere, some country somewhere, they might be trying things like this already. And who knows if this is ethical, and who knows if it might be made illegal in the future. I'm not sure if it's illegal yet. One thing that isn't illegal, there's, I don't know, this is kind of making designer babies, but there's an article, 2 articles in the notes on designer babies. Now, these would be individuals who, let's say they have dwarfism. You have 2 individuals, male, female, who have dwarfism, and they want to produce a child. They want their child to have dwarfism also. Now, there's a form of dwarfism that is caused by a dominant allele. So if 2 individuals who have a dominant allele for dwarfism have a child, there's a 1/4 chance that the child will not have dwarfism. And they might want to have a child with dwarfism. So this is sort of a technique they could use in vitro fertilization to ensure that they have a child with dwarfism. And I believe that's what the New York Times article is about, if you click on that link in the notes to Designer Babies. Again, I think this is legal. I'm not really sure. The way the article is written suggests it is legal. And if this is legal, then this should probably be legal too at the same time. But what we'll cover next is something that's not legal. It's not legal anywhere yet. And that's, I guess that is making specific changes or edits to the embryo. So, one thing I wanted to mention is I want to plug a movie here. So, this thing about SNP profiling and genome sequencing, looking at the genome of the embryos to predict, say, the phenotype that parents might prefer for their children. There's a movie called Gattaca. I think it was released in the 90s, starring Ethan Hawke, where they use this sort of technology here to choose their children. And it's a good movie. It's one of my favorites. One of my favorite movies. And they mostly cover a lot of the ethical implications of a future where this is possible. Okay, so now what I wanted to talk to you about is, okay, so what if a doctor or a scientist or parents, or maybe all three of these get together and let's say they wanted to engineer a human, so edit a gene in an embryo. How would they do it? Well, they could do it using in vitro fertilization. Well, a specific form of in vitro fertilization called ICSI for let me grab a new pencil here, intracytoplasmic sperm injection. Okay, so that's the case. So with this one, what they do is they take the secondary oocyte and you can completely inject the sperm into the secondary oocyte. And when you do that, if the scientist's or physician's goal was to edit a gene, you could insert the Cas9 protein of the CRISPR-Cas9 technology of the type 2 CRISPR-Cas system, along with guide RNA to mutate a gene. Let's say the goal was to mutate a specific gene because you think there's a gene inside humans which is not good. We need to mutate it. So that guide RNA would be specific for guiding Cas9 to that gene that should be mutated. So the guide RNA for a target gene. And this is how one would do it. And rumors are that a scientist named Dr. He Jiankui of Southern University of Science and Technology of China has used this to make the first genome edited humans, the first CRISPR humans. And what he did is, or what he wanted to do was mutate a gene called CCR5. Now, CCR5 is a protein that's found on the surface of certain immune cells. And HIV uses, so this is the virus that causes AIDS, so HIV uses CCR5 to get into the cell. So if humans lack this protein on the membranes of, I think it's white blood cells, then they're resistant to HIV. So his goal was to create humans that lacked this gene using CRISPR technology. And okay, what else can I tell you about this? So I'm going to say rumors here because his research was never published. And I'll talk more about why in a moment. But he told the Associated Press that he created 22 embryos, probably by this method, intracytoplasmic sperm injection, with Cas9, with a guide RNA for this gene. 16 were found to be edited or mutated for CCR5. Some were homozygous mutants, some were heterozygous mutants. Of those 16, 11, and how did he genotype? He probably genotyped with this method over here. Ooh, one thing I wanted to point out is this method of genotyping using a blastomere at the 16 cell stage to genotyping is called what is it? A pre-natal genetic diagnosis, PGD. Okay, so we probably use PGD to genotype these. He used 11 embryos in 6 implant attempts, and 1 worked and resulted in twins. Now I should say that he used 2 groups of parents, and this is one group of parents we're talking about where the father, or the male, was HIV plus, and the mom, the female, was HIV minus. So in some ways he can ensure that the infants produced with this technology, if it works, will not inherit, will be resistant to the HIV virus. So, okay, 1 worked, resulted in twins. And of the twins, let's say twin A was homozygous null for CCR5, and twin B was heterozygous. Okay, so here we have it, CRISPR humans, rumored to be alive and well, somewhere in China. So this came out about 2 years ago. It's also rumored there is a 3rd child born to the 2nd group of parents. We don't know much about this child. When this happened, when Dr. He talked to the media, you can imagine there was an enormous outrage, or maybe you can't imagine, maybe you don't know why there would be outrage. So it turns out this is a really dangerous experiment for a number of reasons. So this CCR5 gene, so humans need CCR5 to resist other diseases. I guess it's key in resistance to malaria. Okay, so sure, these kids are going to, at least the homozygous null child should be resistant to HIV, but apparently they will be very susceptible to malaria. Probably more importantly, unintended mutations. So injecting Cas9 and a guide RNA into an early stage embryo or into at the zygote stage, scientists don't know if that Cas9 is going to make mutations at other locations. CRISPR technology has been used over the world in thousands of labs, and scientists are only now starting to understand the unintended consequences of using Cas9 in these, they can call these secondary site mutations that can occur. So it's not known if Dr. He's experiments caused unintended mutations in these children's genomes. Third reason why there's outrage, he kind of did it secretly, semi-secretly. According to most people, it looks like he did it with, just a few helpers. There are others who think there was a giant conspiracy and there was a lot more people involved. And essentially, Dr. He is taking the fall. So definitely seems to have been done without approval of the government regulatory agencies. And I say all of this is rumor, and Dr. He's talking to the media, and he hasn't published the results, and we know very little about the children resulting from this experiment. We do know that Dr. He, as of December 2019, he was given a 3-year jail sentence. So is Dr. He in jail now? Where is he in jail? You can imagine it's really hard to get information like this. This happened in China for Chinese scientists, and with I guess maybe we're told as Americans that China has strict control over its media, and so you can imagine we have very little details about whether Dr. He is he actually serving the sentence. One of the things reasons why we think the children are currently healthy is that, and I think I read this in an AP report or something, is that this is the maximum sentence you can get for an unapproved medical procedure, assuming no harm was done. So if harm was done to the children, so if the children start getting sick or they end up being injured as a result of this experiment, I think the maximum sentence is much, much higher, or the sentence will be much, much higher than 3 years. So I think that's a good place to stop right there with CRISPR-Cas9 technology. So we know Dr. He kind of, who knows why he claims he did it to, help cure or rid the human population of HIV. A lot of scientists think, he just wanted to be the first scientist to create a CRISPR human. So who knows? Who knows whether or not this was a good idea? It seems like it was not a good idea. It seems like it was ill-timed, and we needed more research before anyone should ever try something like that. Should have definitely had it approved by government agencies and had lots and lots of people involved and considered the ethics of this type of experiment. But okay, so that's a good place to end.